Good morning and welcome to Crop Talk 2020. My name is Allie Noble and I'm the Crops Extension Specialist out of the Prince Albert Regional Office. Each year we hold an event called Crop Talk in partnership with the manager at the Conservation Learning Center, Robin Logan. Originally this was planned to be an in-person meeting in Prince Albert, but due to COVID-19 concerns we have moved it to an online platform. For the online version of Crop Talk 2020, we have a series of 3 speakers that were intended to be at the in-person meetings, joining us via webinar instead. For this webinar, we have our keynote speaker joining us, Jeff Schoenau, to talk about micronutrients. Dr. Jeff Schoenau is a professor of soil fertility and professional agrologist who works in the Department of Soil Sciences at the University of Saskatchewan. He holds the Saskatchewan Ministry Agriculture Soil Nutrient Management Chair in the College of Agriculture and Bioresources and is the Fellow of the Agriculture Institute of Canada. With that, I will hand things over to Jeff here.
Thanks, Allie. Good day, everybody. I'm going to talk to you folks a little bit about micronutrients in crop production. And I'd like to acknowledge the graduate students, postdoctoral fellows, and colleagues that have worked with me over the last little while that I'm going to be referring to research work that they have been conducting related to micronutrients in my So thanks to all those folks that are listed there on the author list. Certainly when it comes to essential and therefore no less important in plant nutrition than the macronutrients like nitrogen, phosphorus, potassium, or sulfur. And indeed, the micronutrient elements like copper, for example, are required in only a few grams per hectare. But nonetheless, they're still considered to be essential in the overall plant nutrition and functioning. When we're thinking about micronutrients that are of interest in Western Canadian crop production, that listing includes copper, zinc, boron, manganese, iron, and chlorine, and those are the micronutrient elements that I will be speaking about in my presentation. Micronutrients, I guess for lack of a better word, I think they would be considered to be rather mysterious by nature, almost rather ghostly. And that's because the deficiencies may appear and then sometimes for a variety of reasons disappear. And the symptoms are often easily disguised, I guess you might say, confused with other forms of stress. And sometimes we have a particularly unique combination of soil, environmental, and crop conditions that's needed for that micronutrient deficiency to in fact show up. And so what that all boils down to, folks, is that it means that it's inherently rather difficult to conclusively diagnose and predict responses to micronutrient fertilization. It's kind of the nature of the beast, so to speak. And also we have, in terms of responses, as I'll talk about, to micronutrient fertilization, often they're small, fleeting, and variable. And as I've said, certainly difficult to predict because of a wide variety of factors that will influence the degree to which a response to fertilization may be observed, if any. Before I go on, I'd like to talk a little bit about biochemistry about micronutrients, just a little bit. First off, the plant absorption of micronutrients. We're thinking about micronutrient metals like copper, zinc, manganese, and iron. These micronutrient metals are absorbed by roots as cations, those are positively charged ions, but they get to the roots as chelates. And what a chelate is it's a metal that is complexed or reacted with a soluble organic matter to form a kind of ring structure that keeps those micronutrients in solution and enables them to, I guess you might say, roll along, move to the root, and where they can be taken up by the root into the interior of the plant. So the chelates stay in solution, and that really helps keep that micronutrient moving to that root where it can be absorbed. Boron is taken up as boric acid, chlorine as the chloride anion, and borate and chloride as anions are actually quite mobile in the soil, and they'll move quite readily with water wherever water is moving in the soil. In terms of uptake of the micronutrients by plants, the main uptake takes place through the roots, and certainly in a soil, that's the dominant uptake mechanism. But we can also have uptake of micronutrients through foliage as well, and that's why for some of the micronutrients, a foliar application is also an effective way of getting that micronutrient into the interior of the plant, where it can participate in those physiological functions where it's needed. And so I guess that brings me to a little bit of plant physiology. What role do those micronutrients play in crop nutrition? So my first grouping there of copper, zinc, manganese, and iron, these are metal micronutrients that are involved in a number of important processes: photosynthesis, the activation of enzymes, hormonal functions in plants, and may also play a role in some circumstances in disease resistance. And a good example of this is that copper deficiency can aggravate ergot infections by causing the floret of a cereal to remain open longer. And I'm going to talk a little bit more about this in a few minutes when I talk specifically about copper. Boron is a micronutrient element that plays an important role in extension of the cell wall, division at the growing points at the meristem. It's involved in pollen tube elongation, and therefore is especially important in reproduction. The pollination process and deficiencies of boron can certainly have negative impacts on seed set and seed development. Chloride, as a micronutrient as well, is involved in balancing of charge in the plant, osmotic water relationships, cell turgor, and it's another example of a micronutrient that does play a role in some instances in susceptibility to disease, and seems to be important, is particularly in cereals, in imparting a resistance to root and leaf diseases. When it comes to identifying a micronutrient deficiency, we can look at a plant, and in the case of copper deficiency in cereals here, I provide you with a picture here, so a couple of photographs. Copper deficiency in cereals can produce somewhat characteristic symptoms, in particular this twisting or pigtailing of the tips of the leaf that is shown there in the case of wheat and also in canary seed. Sometimes and oftentimes these deficiency symptoms are rather non-specific. In the case of zinc deficient wheat here, I show this picture here that shows in a sea of rather dark green, zinc deficient wheat plants showing a characteristic lighter green color, smaller leaves. Sometimes you'll see chlorosis showing up in between the veins. For some micronutrient deficiency symptoms, those symptoms can look similar for different micronutrient deficiencies. A good example of this is shown here: sulfur deficiency on the left-hand side, boron deficiency on the right-hand side, and both of those producing some issues, some problems with seed set, as we can see there. Not too many pods there, small pods, symptoms showing up, some leaf curling there at the on the young parts of the plant, the upper parts of the plant, so that certainly a visual look alone can be not very conclusive in being able to separate out what the problem is. So when it comes to diagnosing micronutrient deficiencies, using a visual inspection alone, as I pointed out, can be risky, inconclusive for a variety of reasons. Those deficiency symptoms can look similar for different deficiencies of a nutrient, can be easily confused with other forms of plant stress, environmental stress, or even things like injury from pests or from, for example, a herbicide injury. So to that end, we have some other tools that are available to us. Soil and tissue testing, I think, are useful tools. Certainly the micronutrient tests do receive a fair amount of criticism and debate, but overall, I would say, as an attempt to measure what's biologically significant in the soil, I would say that in many instances they're no better or worse than a lot of the macronutrient tests. And really, we need to recognize that there are a lot of other sources or reasons why a test may be inconclusive or have a lack of predictive ability apart from the actual functioning of the test itself. And one of those big factors, and I've talked a lot about already, is environmental conditions that can influence whether or not you're going to see that deficiency. But also we have to be aware of variability that exists out there in the field. And micronutrient availability indeed can vary greatly across farm fields. So in order for your assessment, your soil assessment, or even your plant tissue test assessment to be a reliable indicator of what exists out there in the field, you need to make sure that the sampling is done properly. And you really need to know where to look and identify these areas and hone in or zero in on them and do some testing inside the area and outside. Deficiencies in micronutrients often tend to be patchy, tending to occur in patches or localized areas within a field. A good example of that, as I show there in the picture, would be those hilltops, those eroded knolls where we have calcareous conditions, high pH, also sand and gravel lenses that may exist across the field. You might not even be able to observe it from the surface, but if you dig down a little bit, find those types of conditions in the field, soil conditions that are conducive to having a lower availability of a micronutrient. So really your sampling strategy needs to account for this. So that brings me to the topic of soil conditions contributing to micronutrient deficiencies. What kinds of conditions in the soil are contributing factors? Texture of the soil is an important one to consider. Sandy texture indicates a low content of minerals that are capable of releasing micronutrients by weathering, and also a sandy texture generally means a low ability of the soil to hold on to those micronutrients as well. So that minerals that oftentimes are responsible of releasing and hanging on to those micronutrient nutrients are located in the clay-sized fraction of the soil, so that if we have sandy texture, generally means a low release potential and also a low ability to hang on to many of the micronutrients in the soil. Calcareous soils that have high lime content, high pH, generally, the micronutrients are less soluble, less available at high pH. Also, if we have a lot of lime or calcium carbonate present, that will tend to fix micronutrients, particularly copper and zinc, into insoluble forms that the plant cannot readily access. Very low or very high organic matter contents can also affect micronutrient availability negatively, depending on the micronutrient. For example, low organic matter can contribute to low boron availability. On the other hand, soils that are very high in organic matter and contain very little in the way of minerals like clays that can release these metals through weathering, those types of soils like peaty soils can also suffer from deficiency in copper, zinc, and manganese. We also need to think about nutrient balance when we're thinking about micronutrient fertility. High available soil phosphorus, for example, adding large amounts of phosphorus fertilizer to a soil that is maybe marginal in zinc or copper to begin with, can further aggravate that micronutrient deficiency and result in a deficiency of that microelement like zinc, for example. On the other hand, we can go the other way, and some recent research work is showing that addition of copper and zinc to phosphorus deficient soils without phosphorus fertilization can also have an antagonistic effect on plant growth, and particularly this has been observed in cereals. In terms of general soils where we need to perhaps pay the greatest attention to potential for micronutrient deficiency, excuse me, sandy gray, peaty soils, those are the kinds of soils, for example, that we may find particularly in the northern agricultural fringe. Those are the soils that traditionally have had the greatest frequency of micronutrient deficiency. Just to show you some of our recent research work looking at availability of copper in a number of different soils that were collected from across southern Saskatchewan. This diagram shows the inverse relationship between supply of available copper and texture, sand content, and showing here that high supplies of copper in the soil were generally associated in these 14 soils with a lower sand content. So lower sand content, or in other words, higher clay content, equating with higher availability of soil copper, as was pointed out earlier. That being said, generalizations about where and when a micronutrient deficiency will occur, I think, can be a little bit dangerous. So here we come back to relying on our tools that we have available to us, our soil and tissue testing, certainly tools that add resolution. There are some debates about critical levels. Does that level of part per million in a soil test extraction or supply rate, in the case of a measurement of flux, does that represent a marginal or sufficient level? There's some debate about what's actually the cutoff between sufficiency and deficiency. But I think that really reflects the fact that oftentimes, with these micronutrients, the responses are sometimes isolated and not really very clear cut. And that makes it inherently difficult to establish specific guidelines for recommendation as to sufficiency versus marginal versus deficiency. So to that end, I think to really nail things down, use all the tools that you have available to you. A combination of soil and tissue testing, plus some test strips, I think is probably the most conclusive way to address whether or not you're likely to see a response to that micronutrient fertilization. So now I want to move on and talk a little bit about specific responses to micronutrient fertilization for the different micronutrient elements. And I'll start out with copper because of the micronutrients, copper I think is the element that is probably most likely to arise as a micronutrient limitation in Saskatchewan. Certainly on the west side of Saskatchewan and indeed moving into Alberta, copper deficiencies do become more prevalent as we move further west. And when it comes to crops, cereals, especially wheat, are the crops that are most susceptible to copper deficiency. Again, showing some symptoms there in those two pictures there, photographs showing the pigtailing or pinching off of the leaf tips of cereal, and also a little bit later on seeing the empty heads in the wheat associated with copper deficiency. Sometimes a melanosis or browning will also show up on the heads as well under conditions of copper deficiency. Going back to some earlier work that was done by Dr. Mali in East Central Saskatchewan, a gray luvasolic soil near Porcupine Plain, showing here in particular compared to the control, a foliar application producing a significant yield increase in the wheat at that site. Also some other work done by Pat Flayton around the same time, finding foliar application to be an effective means for correction of copper deficiency in the year of application. Some of our work also showing both foliar and soil applications to be effective, and also the soil applications at the rates that would be applied or recommended, maybe 2 pounds per acre to upwards of 5 pounds per acre, for example, of copper applied as copper sulfate, showing a benefit not only in the year of application, but also carrying over and providing some benefit to following crops in rotation in subsequent years. And I'll talk a little bit about that in just a moment. So some work that we recently did, I did with Dr. Ryan Hangs, working in the glass house, looking at copper, zinc, and boron responses in wheat, pea, canola rotation, looking at these responses in a rotational sequence in a number of soils that were collected across Western Canada. So in this research work that we undertook began this about 2015, went across the prairies and collected a number of soils from across the 3 prairie provinces to provide contrast in soil properties, soil conditions, including those soils which we would suspect, based on their properties, their characteristics, their availability assessments, to be potentially responsive to micronutrient fertilization. So these were brought in and used in a number of polyhouse studies. We started in 2015 and picked out 12 mineral soils, where we suspected there would be a potential for response to copper fertilization. And as shown there, indeed, we did see a response to copper fertilization, both foliar applied chelate and salt, and also soil applied copper as well. You will note in this particular trial, we actually saw a negative response to banded chelated copper. And the reason why I think we saw that is we used a little bit too high rate and the chelated form was soluble enough at that rate that we actually saw some toxicity from that particular treatment that we had, which I think points out the importance of recognizing that for micronutrient elements, I'm thinking here of copper and in particular boron, that there is a fine line between sufficiency and toxicity. So we do need to be careful that we're not over applying those micronutrient elements, because depending on the micronutrient, the soil type, we could run into issues with toxicity. In this study, we grew pea after the wheat. And as shown here, we added zinc to the peas in 2016. We maybe saw a trend towards response of the pea to the zinc fertilization, as shown there with the soil applied zinc sulfate, but it wasn't statistically significant at the 5% level. But the interesting thing is that we did see a significant response of the peas to the copper that had been applied the year before to the wheat. And so we're not sure the reason why. It perhaps maybe was a fungicidal effect, because copper sulfate can indeed control fungal organisms. Maybe it had an effect on the fungal populations, the disease organisms in the soil. We're not sure. Maybe it was a nutritional effect. Peas are generally not considered to be particularly responsive to copper fertilization. We did see that trend of pea yield responding positively to soil and foliar zinc application, but it wasn't statistically significant. I guess which brings us into zinc as a micronutrient element. Going back into the 1980s, trials that were conducted, a number of trials by J. Paul Singh back in the 1980s, really didn't see any significant yield responses of dryland crops to zinc sulfate in a number of field trials. Some work that was done in the 1990s along the lines of some precision application looked that there were specific portions of the field, like eroded knolls, that were identified as being potentially responsive to zinc application, in this case cereals. Certainly corn is a crop that does have a high zinc requirement, and there is potential for response there in low testing soils. And again, as I mentioned before, high rates of phosphorus application that are soils that are marginal or deficient in zinc can induce a zinc deficiency. So that balance is always important to consider. And the nature of this mechanism, not really sure why this happens. It may be something related to the phosphorus interacting with the zinc in the soil itself, or how that phosphorus application might affect and uptake might affect the uptake and movement of zinc through the plant. But certainly something to be aware of as well. We did some work with lentils on zinc in the polyhouse and glasshouse studies about 4 years ago. And we did see a few positive responses on brown soils, brown soil climatic zone soils from southwest and south central Saskatchewan. We did observe in our studies that generally the high organic matter soils that we collected and grew lentils on, from dark brown, black soil zone further north and east, we saw limited or no response on those soils. This was an Ardale Association brown chernosem, where we did see a significant response to the zinc fertilization as shown there. But certainly the overall responses in this study tended to be fairly small and variable. Out in the field, a field trial that was done by Sarah Anderson as part of her master's work on a soil in south central Saskatchewan that did have low extractable zinc. We didn't see any response in zinc fertilization despite the low extractable level of zinc in the soil. At the Saskatoon site, where we had high available zinc, we didn't expect to see a response, and we didn't, in agreement with that. The zinc was applied in a broadcast and incorporate application, and what may have happened is that in the soil, we found some evidence of this chemically and spectroscopically as well. When you've got a lot of carbonates present in the soil, as we did at the Central Butte site, particularly in the B horizon, there is the potential for that zinc to react with the carbonates and become rather insoluble. So maybe the reason why the lack of response that we saw there at that site at Central Butte in south central Saskatchewan may have had to do with it being applied as a broadcast and incorporate application, which tended to increase the potential for fixation of the zinc by the carbonates. And we may have seen better results there if it had been placed in a band. Some positive trends for responses of P to zinc fertilization in a glasshouse study that was done a little bit later on by Noah Rahman in his PhD work, and also some work that was done on another group of soils, but was really only significant for one organic soil. This was a work that we did on a soil from southwestern Saskatchewan, a heavy clay soil from Scepter area, Scepter Association. We did see a trend there for the response of P to zinc, but it wasn't statistically significant at the 5% level, but there was a trend there, particularly for the soil applied. For the wheat, interesting, this was a site where we maybe didn't expect to see a response to copper fertilization based on the soil test level, but actually we did see a significant response to copper fertilization of wheat on this particular soil. The other thing that we looked at was how the zinc fertilization affected the concentration of zinc in the grain. And this is important because from a marketability standpoint, especially in the parts of the world where we sell our pulse crops like peas and lentils in the Middle East, they do value grain that is of high zinc concentration, high bioavailable zinc, because there's a fairly high incidence of zinc deficiency in the populations in some countries in the Middle East. And so we can see here that the zinc fertilization, particularly the chelated form, did increase the concentration of zinc in the grain. Moving on to boron. When it comes to boron deficiencies, canola, alfalfa are the crops that are identified as most susceptible to deficiency. Looking into the literature, large yield responses to boron fertilization that are reported in literature are fairly rare. Some of the early work that was done back in the early 2000s, studies showed even on soils that had very low extractable boron content, some of the studies reported no significant yield response of canola to the boron application. And some of our work that we did in our 12 mineral soils where we suspected that there may be potential for micronutrient deficiency, only 2 of those 12 mineral soils responded positively to boron fertilization in our glasshouse research work. This is some of the data from that glasshouse study that was conducted by Ryan Hangs in 2017, and we can see some trend there perhaps towards some response to boron in some of the soils, but it was really only significant on 2 of the 12 mineral soils. Those soils were sandy soils, low organic matter content, low in available boron, so they did have the conditions where we expected to see a response, and we did on those 2 soils. Again, the interesting thing is that residual copper from 2015 did produce a canola yield increase in 2017. So there is again some potential for carryover of effects of micronutrients availability increases from previous years' fertilization to perhaps have of some positive effects in subsequent years. This was one example of a soil in which a PhD student, Noah Rahman, did observe a very significant and large response to boron fertilization. This was a soil of the White Fox Association collected from around Nipawin, Saskatchewan. It was an orthic dark gray Chernozem where we did see a particularly large response as shown there to soil applied boron. The foliar applied boron, for whatever reason, and we had it applied at a couple of stages, it didn't seem to be very effective. In other studies, the foliar applications of boron have proven to work well, and maybe it was just the timing that we applied that foliar that wasn't the optimal timing in this particular study that we did. Manganese. Just a few words about that. Cereals, especially oats, growing on highly peaty soils in the northern agricultural fringe could potentially respond to manganese. Generally would say deficiencies of manganese are rare in mineral soils. Something that's come up in the last 3 or 4 years, isolated incidences of manganese toxicity. This was identified by Lyle Cowell, agronomist with Nutrien, working and observing some deficiency or symptoms in the plant that in canola plants and also in cereal plants that pointed towards manganese toxicity. These were very sandy, acidic gray soils in east central Saskatchewan. And I had a fourth-year thesis student who took some of that soil that Lyle had collected, and we grew canola on it, and we could see the kinds of deficient or toxicity symptoms, I should say, that were kind of reflective of manganese toxicity, some bubbling and necrosis on the edges of the leaves. And when we added lime, increased the pH, the canola dry matter yield there that we measured in pots that were amended with lime nearly doubled. And we also saw a decrease in the tissue concentrations of manganese from toxic to adequate. So pointing towards, in this particular case where it was identified, a beneficial effect of liming, decreasing the solubility of that manganese and lowering it from toxic levels to levels that were non-toxic in the soil. Iron, I will say just a few words about iron. I probably wouldn't be talking about iron at all, except for the increased acreages that we've seen of soybeans on the prairies in the last few years. Iron deficiencies produce rather marked symptoms in the plant. Iron deficiencies produce a condition called iron deficiency chlorosis, where you get this chlorosis or yellowing in between the veins there, as shown on the pictures there, one from the field and another a plant there. In soybean, on the young leaves there at the top, this rather pronounced yellowing in between the veins. And there's a number of soil conditions that kind of combine or can come together to result in this iron deficiency chlorosis. High pH is associated with lower solubility of iron compounds in the soil, which reduces availability. Poor drainage can also aggravate this as a result of the buildup of carbon dioxide in the soil and the accumulation of carbonates. Carbonates, nitrates, salts in the soil also aggravate iron deficiency. And soybeans happen to be rather inefficient users of iron, and that's why IDC tends to show up most in soybeans, and some varieties are more sensitive to iron deficiency than others. And there are, for different soybean varieties, iron deficiency chlorosis ratings available. And if you anticipate conditions that are liable to result in iron deficiency chlorosis, you can look and choose a variety that has a greater resistance to this condition. Genetics have been developed here to able to at least partly, and in some cases I think do a very good job of overcoming this iron deficiency chlorosis in the field. We did a study looking at response of soybean to iron and a field trial in southern Saskatchewan, where we had soils that had the kinds of conditions that were conducive to iron deficiency chlorosis: high pH, a little bit saline, high nitrate content on lower slope position that would be conducive to some potential flooding and saturation that will aggravate this iron deficiency chlorosis syndrome. Worked with two soybean varieties in this trial, one that was IDC sensitive and one that was IDC tolerant. And what we found the first year that we did this, we had the soil conditions that were conducive to iron deficiency chlorosis, but it was a dry spring. So we didn't have any of that soil flooding that would produce those conditions of high buildup of CO2 and carbonates that interfere with iron nutrition. So we didn't see any response to iron fertilization in that first year. However, 2016, a similar soil, but here we had some heavy rains in June that produced some flooding, and we did see IDC develop. And IDC did develop in the sensitive variety, and we did see a significant response to foliar iron application. Whereas the IDC tolerant variety, which we expected, did not respond to iron. It didn't show any symptoms, and it also did not respond to iron fertilization, understandably. And so this is with our susceptible variety of soybean here. showing particularly some trends of response to other forms of iron, but the iron chelate that was foliar applied did produce a significant increase in the yield of the soybean that year. Now, this is 1 year, one site, so we have to recognize the limitations of being able to extrapolate those results to other sites and other conditions out there. I think overall genetics is probably your best defense when iron deficiency chlorosis is a concern. But at least, again, based on only one site and 1 year, we did see a foliar iron may be a suitable rescue treatment when the iron deficiency chlorosis does show up. Last micronutrient I'll talk a little bit about is chloride. There is potential for responsive cereals in highly leached soils with low chloride levels in the soil profile. Generally less than 30 pounds per acre of chloride in the top 2 feet has been used as a kind of diagnostic criterion for this traditionally. Was first identified, a lot of work back in the 1990s with winter cereals showing potential for response of winter wheat to chloride fertilization in some of these soils that have very low contents of chloride in the profile. The response may be associated with disease pressure. Some work that I was involved with a few years back where we looked at responses of this case, hard red spring wheat to chloride applied as potash in the landscape in both upslope positions and lower slope depressional positions. We did see 1 year a significant yield response of wheat in highly leached depressions to the application of 40 pounds of KCl, which we attributed to a response to chloride because we did have very low contents of chloride in that soil that year throughout the profile in the spring of the year. The next year, it was a dry year, and the chloride levels were back up again in the profile, and we didn't see any response to the fertilization of chloride that year. It might have had something to do with disease. Unfortunately, we didn't assess the disease incidence in this trial. I would say overall, probably that since growers are tending to use more frequently potash in their blends for their different crops, has probably reduced the incidence of chloride as a limitation compared to what we were seeing maybe 20 or 30 years ago. And the one thing about chloride is that nearly all of the chloride is contained in the plant in the straw. And what that means, the straw is retained on the field, that chloride has an opportunity to be recycled and reused. It does move a lot in the profile. It can be leached down deep. So probably the place to look for potential chloride deficiencies are conditions where you have a lot of leaching, a lot of movement of water through the profile, as in depressional areas in the landscape, and also sandy textured soils. When it comes to responses of crops to chloride fertilization, canary seed has been identified as a crop that can be particularly responsive to chloride addition. Fourth year thesis study I did Kirsten Thieker a few years ago, showed some responses of canary seed to the addition of KCl that was attributed to an effect of chloride. And also Bill May at Indian Head Agriculture Agri-Food Canada showed seed yield increase in one case of about 24% from chloride that was added to canary seed. This was at a site in the dark brown soil zone. So that's a crop where particularly as well there is a potential for response to addition of potash and that potash being a source of chloride that may provide benefit. So some final thoughts here, folks, to wrap up. Micronutrients certainly are something we need to think about as part of the overall balance of nutrients that is required to optimize our yield and economic return. And also, I think, to be thinking about them both in the short term, that is this season, and also over a number of years in the rotational cycle, as micronutrient fertilization addition can carry over, particularly for the soil-applied micronutrient fertilizer salts, can carry over into subsequent years and could potentially provide benefit to those following crops. Micronutrients certainly do become a more important consideration when you're aiming for the top of the yield curve and trying to get every last possible bushel of per acre of yield that you can get out of that crop through management. The patchy and variable nature of micronutrient deficiencies makes it challenging in terms of getting a representative sample out of there, but so we need to think about a careful soil sampling strategy, but also makes them an obvious target for precision fertilization, identifying those areas and certainly then applying micronutrient only to those parts of the field that needs it. A multiple evidence approach, I think, is the best way to identify and verify deficiency. So using all the tools that are available to you, your soil tests, your tissue tests, your test strips, I think is going to be your best approach to trying to nail down as best as possible the potential for seeing an economic response to fertilization. But certainly overall, the complexity of factors that out there, environmental variability factors out there that affect that micronutrient availability and nutrition certainly inherently makes prediction of likelihood of response a challenging endeavor. And indeed, thinking about the ability to predict, certainly the ability to predict that upcoming growing season weather is always a challenge in making fertilizer recommendations for any nutrient out there, whether it's a micronutrient or a macronutrient. But we want to use the tools and our understanding of factors affecting those responses as best as we can to manage our micro and macronutrient decisions effectively. So with that, I will conclude, and thank you folks for your attention.
Thank you for that wonderful presentation there, Jeff. At this time, I would like to thank everybody for taking the time to join us for this webinar, and a special thank you to Jeff for being flexible with the current schedule we're working with and being able to give us a webinar instead of an in-person presentation. You can easily find the recordings of our other previous webinars that we have put on with our crops group by searching for webinars on our website. For more information on everything crops related, you can go to www.saskatchewan.ca/crops. If anyone has any questions regarding this topic, please feel free to email us, as this is a pre-recorded presentation. We won't be having live questions. If you have any questions regarding anything else farming related, please contact your local SaskAg regional office or the Agriculture Knowledge Center at 1-866-457-2377, and we will be sure to put you into contact with a specialist to help you answer your questions. Even with everything going on, all the specialists are working from home, so we're still easily reachable. Thanks again, stay safe, and have a great day, everyone.